Good morning, good afternoon, good evening, wherever you are. Welcome to this webinar on pediatric cure. I'm Lynn Moffinson. I'm the Senior HIV Technical Advisor at the Elizabeth Glaser Pediatric AIDS Foundation. Next slide, please. So what we're going to do today is we're going to have a brief introduction from me on why cure research is here. And then Debbie Prasad will give us an overview of what is PAVE, a global landscape of the goals, membership, and the trials, pediatric cure trials. Dr. Mark Cotton from South Africa will talk about broadly neutralizing antibodies and animal models. And then we will have Josephine talk about youth and the movement towards cure. And Martha will talk about, as a community advocate, reflections of mother and long-term pediatric cure. And then we'll have a discussion, which I'll moderate. Next slide, please. A few housekeeping rules. Everybody is muted automatically, but all are welcome to engage at any point during the webinar. And let me point you towards at the bottom of your slide, there is a Q&A box, question and answer. And please feel free to ask questions through the Q&A box, and we will try to get to all the questions at the end. This is being live streamed and recorded, and it will be shared. And if you have any connectivity issues, please chat with Sarah or Cassette through the chat box or email. Next slide, please. So pediatric cure research is here. Next, please. So why is now the time for pediatric cure? What's new since the Mississippi baby? Next slide. So first, several additional cases of HIV remission with very early treatment and perinatal infection have been reported. The first on the left is from France, and the second is from the SHARE study in South Africa, raising the hope of post-treatment control after perinatal infection. Next. So very early treatment at the time of diagnosis is now standard of care for newly diagnosed infants and children with HIV. And we know that very early treatment in infants is life-saving. And this is a slide from the SHARE trial showing us a 75% reduction in mortality with early treatment. And now this is recommended by the World Health Organization, by the US guidelines and other guidelines for immediate treatment as soon as infants are diagnosed. Next slide, please. HIV diagnosis at birth is feasible and being implemented even in low and middle income countries. And studies by EGPAF and others have shown that point of care early infant diagnosis is feasible, acceptable, and cost effective in low and middle income countries, as you can see from these studies. And point of care is now recommended for infant diagnosis by WHO for a diagnosis in infants and children younger than 18 months. Next, please. HIV treatment at birth is feasible and also being implemented in low and middle income countries, as you can see from a number of different papers from African countries, including Malawi, Kenya, and a study from 6 countries. And additionally, more potent antiretroviral drugs are becoming available for newborns, including raltegravir, abacavir, and hopefully dolutegravir. Next, please. And we know that very early treatment reduces the viral reservoir. And you can see on the left a number of different slides that have demonstrated this. And on the right, you can see a slide from the early infant treatment study. And the little colored dots are infants who started on treatment within 24 hours that have been followed through 96 weeks. The squares show you control infants who started ART at a median of 4 months. And then the little triangles are adults. And you can see the decline in cell-associated DNA over time in the early treatment, and that the cell-associated reservoir is much lower than children who start early at 4 months or in adults on long-term treatment. Next, please. Immune adjuncts to treatment are being studied in neonates and infected infants, and Mark is going to go over this in more detail. including he will talk about the recent study coming from Botswana from Roger Shapiro and all, where they gave 2 broadly neutralizing antibodies IV every 4 weeks and showed maintenance of viral suppression. Next, please. So since the Mississippi baby first demonstrated that very early treatment can result in significant treatment-free remission of HIV, very early diagnosis and treatment of infants has become standard of care globally. Early treatment, even with the currently available lower potency regimens for newborns, have been shown to significantly reduce the latent viral reservoir. New and more potent treatments are becoming available for newborns, and immune adjuvants to treatments such as broadly neutralizing antibodies have already been shown to be safe in neonates and young infants. Next, please. So it's time to harness the changing treatment landscape of very early treatment, take advantage of the unique viral and immune aspects of perinatal infection and the results that we've seen from very early treatment studies to begin to accelerate research on assessing and optimizing immune-based strategies for remission and hopefully cure in children with HIV. Next slide, please. So Dr. Persar is now going to talk to us about PAVE. And Debbie, it's all for you now.
Thank you, Lynn. Next slide. Good morning, everyone, and thank you, Lynn, for that introduction. So PAVE stands for the Pediatric Adolescent Virus Elimination, Martin Delaney Collaboratory. Next slide. I wanted to start with a tribute to Elizabeth Glaser and the foundation. Next slide. And as most of you know, Elizabeth was instrumental in starting advocacy for treatment of pediatric HIV infection through our own experience with infection in her 2 children. And I want to point you to the text in the bottom of the slide on page 66 of her book, in which she said, maybe this won't all end tragically. Maybe they will find a cure or at least a stabilizing drug. And I think at that time, Elizabeth lived through this epidemic from the 1980s to the 1990s. She had a vision for cure. And now in 2021, with the initiation of the PAVE Martin Delaney Collaboratory that specifically focused on HIV cure, we've come a long way. But for myself and my own journey, this was a time for reflection in presenting today to this group. And I myself was a recipient of an Elizabeth Glaser Pediatric AIDS Foundation Award in 2005, that was actually specifically focused on studying viral reservoirs in children. So these are cells that maintain HIV in a quiescent state that prevents eradication of these cells. And through these studies, it's actually led to the tools and methodologies to identify the case of the Mississippi baby in 2013 that was celebrated at a foundation event where I met G. Glaser. That's been very inspiring to me. Next slide. And so to start with the framework for HIV cure research and the foundation behind cure research, as most of you know, antiretroviral treatment is really highly effective in sustaining HIV replication to undetectable levels for years, even decades. We know that we have many more steps to go to get our pediatric formulations to a point where our kids can achieve this goal of 90% achieving suppression. But this sustained suppression does not lead to virus eradication and cure, largely because of early establishment of long-lived infection in memory CD4 T cells, where intact proviruses persist in a non-expressed quiescent form, and therefore cannot be targeted by ART or host immune responses, and permits a lifetime of viral persistence. So the goal of HIV remission and cure therapeutics is really discovery, as Lynn alluded to, discovery of effective immunotherapeutics that would circumvent lifelong ART, its toxicities and stigma of living with HIV. Next slide. And just to kind of highlight visually what this looks like is this reservoir shown in different colors on the far right. It's really comprised of a heterogeneous population of cells that really survives for an individual's lifetime. And the goal of HIV cure therapeutics is to dramatically reduce the amount of these cells persisting in an individual and identify ways to reactivate the virus in this latent state. Next slide. What you can see here are early seminal studies in 1999 showing that when ART is stopped, there's prompt rebound in viremia within 2 to 4 weeks of stopping antiretroviral treatment. And so the goal of HIV cure and remission therapeutics is to delay this rebound time on the order of years to hopefully a lifetime. Next slide. And so this is why we were poised to recognize that the case of the Mississippi baby was really unique and different. And what I hope you can see on this slide is this infant with clear infection as showed by closed circles with detectable HIV infection achieved 27 months of remission, but then developed prompt rebound. So our goal here is to delay rebound by on the order of months to years rather than weeks. Next slide. And so the PAVE Collaboratory, the mission here is outlined. We really want to use cutting edge science to establish a deep and broad understanding of the immunopathogenesis of pediatric HIV reservoirs across the age spectrum, and to demonstrate safety and efficacy of novel therapeutics to purge and control HIV reservoirs and SIV reservoirs in preclinical models in non-human primates that would pave the way for future interventional studies towards a lifetime of sustained HIV control of ART. And so our vision here is sustained ART-free control of HIV replication in pediatric populations, different ages, different durations, and that we believe this will be possible with targeted approaches to eliminate these reservoirs combined with long-acting strategies to control viral rebound. Next slide. So a central part of the PAVE Collaboratory is very futuristic. It's really a vector-based technology approach for delivering these broadly neutralizing antibodies using AAV-associated vector that can be given as an injection and hopefully persist for the lifetime of an individual. And what you see here on this slide is an example in a non-human primate model, an adult primate, non-human primate model, where this monkey was given this AAV vector that contained proteins for 2 broadly neutralizing antibodies. And shown in green, you could see continued suppression of virus replication for out to 150 weeks with this broadly neutralizing antibody cocktail delivered intramuscularly. And the inspiration around thinking about this technology for the PAVE Collaboratory was really built on recent study. This is not so futuristic in adults, where these are phase 1 studies looking at safety of AAV delivered VRC07 in adults as pioneered by the VRC John Mascola and Joseph Cazaza. Next slide. And so our Martin Delaney Collaboratory is 1 of 10. I want to point out that this is with pediatric PAVE as the only one, and with large investments from the NIH, $53 million a year towards this goal. So a very serious commitment to HIV cure and remission. Next slide. And who are we as PAVE? I'm really pleased. We've really brought together team science here. People have been working in this field for years. We're about 36 investigators, 17 institutions in 3 continents, including Africa and Europe. Next slide. And there are several project goals. It's divided into 5 focus areas. The first really deals on in-depth understanding of the biology of reservoirs that's largely built around human cohorts. The second is understanding HIV-specific immunity, specifically broadly neutralizing antibodies. The third is identifying in preclinical models how to purge these reservoirs, and then with the hope of moving them into clinical trials. And then more importantly is designing and optimizing assays for quantifying reservoirs on low blood volumes on children. And then importantly, the community engagement in helping us shepherd this research agenda along. Next slide. Next slide. And a unique part of our collaboratory is really we rely heavily on these unique clinical cohorts and biobanks that are global in nature, with collaborations with clinical trial networks, such as the IMPACT and the EPICAL consortium, and within the US, the FACS cohort that's been established for decades here. Next slide. Important to this and to this agenda is really establishment of industry partners to allow us to move novel compounds into preclinical models, ex vivo studies, and then ultimately to clinical trials. And this slide lists several of our industry partners, including Immunity Bio, Inimune, and ViiV Healthcare with novel compounds, innate immune enhancing agents to purge viral reservoirs. Next slide. And so what we hope through this combined approach is an iterative process that will eventually end up with a complete comprehensive picture to understanding HIV and SIV reservoir biology towards remission and eradication, with support from our community program and industry partners and clinical trials enterprises. Next slide. And our community engagement program is, this program is a reflection of that with our collaboration with AVAC and the Elizabeth Glaser Pediatric AIDS Foundation. Next slide. I just kind of want to point out that this is a team science approach with leaders of these different research foci distributed amongst expertise in the field to help bring the science together, lead the science forward, and mentor the next generation of HIV cure researchers. Next. And so what we hope is by the end of this, or hopefully year 3 or so, we identified novel strategies that will inform development of clinical trials and biomarker studies to achieve HIV remission and cure in perinatal infection. Next slide. And so I kind of want to come back to this case in the spirit of this webinar with respect to E to A, evidence to action. I think the next slide will highlight how this single case of the Mississippi baby has brought us to this point. And this strategy of very early treatment that Lynn alluded to has been rolled out in multiple countries in this IMPACT trial, showing feasibility of very early infant diagnosis and treatment with safety data. Next slide. And then I really want to take this time to acknowledge Lynn Moffason for all her efforts in supporting these research endeavors, pathogenesis studies over the years to bring us to this seminal point of really showing that very early treatment reduces the viral reservoir in perinatal infection, the first step towards remission and cure. Next slide. And so why focus on HIV cure research? And this is not unique to pediatrics. We want to prevent new infections. We want to overcome the limitations of antiretroviral treatment, combat stigma and discrimination, and provide a sustainable financial solution for the HIV pandemic control. And this is really a nice review by Mark Dyball and the group at Sunnyland summarizing why focus on HIV cure research at this point in time. Next slide. So I just want to end with being indebted to the PAVE Martin Delaney Collaboratory team. This is really a team effort. I'm representing the group, and we look forward to engaging more partners and moving the science along. Next slide. So with that, I'll introduce Professor Mark Cotton from Tygerberg Children's Hospital and Stellenbosch University in South Africa to give you an update on current trends in data on broadly neutralizing antibodies and animal models.
Thank you very much, Debbie. And I also would just like to state that I was a Pediatric AIDS Foundation scholar in 1995 when I was working in Denver, Colorado, and did laboratory work in Terry Finkel's lab at National Jewish. And I think this exposure gave me some insight into how the laboratory works, and at least to have some idea of what's going on in the laboratory. And I'm extremely grateful for that foundational experience. So to talk about broadly neutralizing antibodies, and if I can have the next slide, this is certainly, next slide, please. So I wanted to go back to the basics again. And the key is how HIV gets into the host cell. And you can see this cartoon. There's it's GP120 and GP41 that undergo conformational changes and enable binding with the co-receptor and entry to the cell. So the HIV envelope you can see is trimers of GP120 and GP41, and an ability to think spatially, I guess, which I can't do, by the way, is essential to working or designing and thinking, strategizing about broadly neutralizing antibodies. Next slide. So the envelope is protected from antibodies. And, firstly, the envelope spikes are very sparse. And, in comparison to coronavirus and influenza virus, which are covered in spikes, there are not that many in HIV, and that protects HIV. There's all of the glycosylation hides conserved areas, and non-functional parts of GP120 and GP41 make decoy antibodies. So it's extremely difficult to develop meaningful antibodies to HIV. Next slide. So, in fact, the search for broadly neutralizing antibodies has been going on for some time. And as early as 1991, in studying asymptomatic individuals living with HIV and doing quite well, that the investigators were able to find meaningful and effective antibodies in these patients. And this was really the start. The methods were quite laborious, certainly more so than now, but the promise of antibodies has been known for some time, thanks to these investigators. Next slide. So where do they act? And there are vulnerable parts of GP120, and I've just listed the names of sites where these broadly neutralizing antibodies actually act, and this is a importance when one's thinking of possibly combining antibodies. Next slide. So how did this field unfold? And as I'd mentioned, the first generation of neutralizing antibodies were hard to develop. And as techniques improved, so one was able to screen many patients living with HIV for antibodies. And in fact, Debbie has already mentioned that one can even deliver antibodies now through carrier viruses. The very small table on the right is just to illustrate that this field is expanding dramatically as larger and larger samples are being screened for antibodies in more and more patients living with HIV, and more of these antibodies are being uncovered. Next slide. So what about the timing of antibody development to HIV? And as in the first weeks of HIV, one develops binding antibodies, then strain-specific neutralizing antibodies, and over time, broadly neutralizing antibodies. The problem to the patients or the individuals in whom these Nabs are identified, it's of no use to them because the virus has already moved on, but it is of use to others who have not had to develop or escape these antibodies. And this also gives us a pointer that in the future, resistance to the antibodies has to happen. Next slide. So children seem to have a greater propensity for developing these Nabs than adults. And one study over here shows quite a difference where round about 75% of children in one study versus 19% of adults develop broadly neutralizing antibodies. And again, it's really advancing HIV that is driving this Nab development. Next slide. So what are the roles in children? Well, it could have a role in treatment. It has a potentially vital role in cure remission work, where at some stage you have to stop antiretrovirals, and the BNAbs may prevent massive rises in viral rebound, and to the benefit of the patients. And then, of course, for children, there's the possibility of vertical transmission prevention, and especially breastfeeding, which is responsible for some continued low-level transmission. Next slide. So what about adult studies? What have we learned from some of the BNAb trials that have been done? First is that BNAbs can suppress viremia in adults with chronic HIV. And here you can see two BNAbs that had some temporary benefit in dropping the viral load in these patients. Next slide. Already they noted resistance, as we've mentioned in the pathogenesis, or how these BNAbs develop. That resistance is unfortunately part of the spectrum. So it's something to worry about. VRC01, another BNAb, has also suppressed viremia. And you can see it was quite good, but the virus bounces back. Next one. So it has been used during ART interruption, and with some benefit. And in this study by Sheed et al., you can see that a single BNAb prolonged the time of being having viral load suppression compared to control. So there's definitely a benefit over there, and that needs to be determined just how much it is. Next one. So already, because of the resistance issue, people are looking at combinations. And here's a study showing that a combination of two BNAbs will give you a longer delay to rebound than a single BNAb. And we are incorporating this type of data into an impact study that we are planning in the cure remission agenda using BNAbs at this stage. Next slide. What about other effects of BNAbs? Well, it's possible that BNAbs actually have a vaccinal effect, so that they could enhance other forms of immune response. And you can see the first slide, and by tier 2, we mean those are viruses that are they're relatively resistant to the first generation of BNAbs. So better humoral responses have been shown, and also better CD8 responses have been shown in the non-human primate model. So there's at least some hope of additional benefits through using BNAbs. Next slide. So what about children, and what has been done in children so far? Next slide. Well, again, there's a use for promoting early virological control and for maintaining virological suppression. Next slide. And we've mentioned the possibility of supporting ART-free remission. So all of these areas are under study, and we hope that BNAbs will contribute to this agenda in children. So what about trials in children? So firstly, I want to mention Impact 2008, that's led by Betsy McFarland. And this study uses VRC01 in conjunction with antiretrovirals in early treated infants. and with the hope of reducing the HIV reservoir. And Debbie has already mentioned Impact 1115, which is the study designed to replicate the Mississippi baby. And it's hoped that BNABs will further drive virological suppression and smaller reservoirs in these children. I will be talking about Roger Shapiro's amazing study from Botswana that was released at CROI, and Philip Golder is preparing a similar study in KwaZulu-Natal, which I won't be sharing data on that. Next slide. So what BNAB studies, what other studies have been done? And there are 2 studies done in newborn infants, and these are exposed uninfected infants. The first with VRC01 that was shown to be safe right throughout breastfeeding, and then using the long-acting form of VRC01, extended safety in a similar group of infants, again, exploring the possibility of BNABs in preventing transmission of HIV, which we really hope will happen. So what about Roger Shapiro's study? It's the Telo trial, and that means the next thing in Setswana. And this study was shown and discussed at the recent CROI meeting in February. And this involved 28 children who'd been treated from less than or equal to 7 days of life. And all of them were very well suppressed in the 6 months before study entry. And the study entry was between 2.4 and 5.6 years. They all had very good CD4 counts. They were brought in for the study. They were monitored carefully. And then 2 BNABs were given weekly ×4. Antiretrovirals were then stopped, and the primary endpoint was the number with an undetected viral load 24 weeks off antiretrovirals. And you can see the 2 BNABs that were chosen act at different sites on GP120. So what did they show? Next slide. So in the 8-week observation on ARTs, viral loads were detected in 2, so they could not continue. And then they did a safety and PK study on 6 children, using the 2 BNABs given 4 weekly times 8 doses. That's for quite a long time. And they stopped towards the end. They stopped the ART for 24 weeks according to their study design. And they showed really, really good suppression, that 83% or 5 out of the 6 were suppressed. In the remaining 20 children who received shorter exposure to the BNABs, 30% or 6 were suppressed after 24 weeks, and they all went back onto antiretrovirals. Next slide. So some concern, I guess, that there does seem to have been some virological rebound, and they will be looking at that more to see if there was resistance, but luckily they're all suppressed again on antiretrovirals. Next slide. So this is just to introduce the non-human primate type of study, which is quite complicated and quite excellent, and pediatric and neonatal non-human primates are being used to inform studies in this age group in humans. Next slide. And the study that I just want to focus a tiny bit on is the triple combination BNAB approach. And because we're having resistance with 1 and possibly even 2, there's an important need to develop cocktails with very broad activity. And the first step towards this is in the non-human primate model. So we're very grateful for this work and hope that it will inform us in the future. Next slide. And then just to thank colleagues for support in preparing this talk. Thank you. And if we could have the next speaker.
Josephine is going to, she's an EGPAF board member and youth advocate, and she's going to give her feelings about how youth feel about the movement towards cure. It's all yours, Josephine.
Thank you so much. I hope I'm heard loud and clear. My name is Josephine Kenya, as I've been introduced, and I'm going to share about what young people feel and what they think about an HIV cure. And when I received this question, definitely I had to categorize the types of young people that I'm going to present today. So there are those that have been informed about a cure. They know about some people that have been cured from HIV. They've heard about some studies happening around HIV cure. And sadly, this category of young people, they've been informed mostly through these international conferences that we go to. So the likes of me, like I had to go for an HIV cure fellow, academy. And that's how I got informed about an HIV cure. The likes of other people that have been informed about the HIV cure have been informed through different trainings that have been held, but these have only targeted a few of the young people that already are working at health facilities. So this category of young people that are a bit informed about an HIV cure, they're excited. That is number one, because I mean, who would love to have the virus for more than 10 years or even a year? Because we've had Corona around and it's just 2 years and everyone is just tired of the virus. Like they want to be cured. They want the vaccine to come. Like they want to be cured. So this category of young people that have known that something is being done around the HIV cure world, they are very excited, but at the same time, they're a bit disappointed. And why are they disappointed? They're disappointed because they think the movement towards an HIV cure has been a little bit slower than expected, especially in this era where we had the coronavirus coming and, within just months, the vaccine was available. So they think they're excited about the developments that is happening around treatment because, you now take lesser pills, we have lesser side effects, the injections are in the pipeline. So they're all excited about these developments because to them, it seems like we are getting closer to the cure. Likewise, the developments around PEP and PrEP. So all that gives hope that actually the cure is coming. So when you get to know even the other studies that are being done and the other people that have been cured cured from HIV, they're excited, but they think the movement is a little bit slower because Corona has just come and just in just months, the vaccine was available. So they're eagerly looking forward towards understanding more about an HIV cure, what's happening, not only in the developed countries, but what's happening within their own countries. Because when you look at the burden, it is mainly in Africa. So I'm happy when I hear about the Mississippi baby. I'm happy when I hear about the other fellows that have been cured about HIV, but there is that excitement that comes when you hear that actually someone came from Africa or someone came from your own country and they've been cured of HIV. So they're excited, but they need to know more. They would need to get more studies happening within their own countries, and they would need to see more people within their countries getting cured, because that then brings in a very, very big hope that the cure is, within their reach. Then the other category of young people is those that have not heard anything about an HIV cure, and all they've had is myths and misconceptions. Now, that kind of category is the largest because HIV cure information is rarely available to all young people or to all people generally people living with HIV. So they feel like the quest for that information is there, but the gap has not yet been filled. For example, when we go to the health facilities and probably there is a curriculum for healthy talks. They'll talk about adherence, they'll talk about stigma and discrimination, they'll talk about disclosure, they'll talk about prevention measures, but nothing will be talked about around HIV cure. Yet the clients in care want to know more about an HIV cure. So this category of young people that are not informed, and all that they hear or all that they know about cure are the myths and misconceptions. And these include things like HIV is a business, so there's no way someone will let this business go away and bring a cure, so people would prefer treatment to cure. There are also myths around virgins cure HIV, so if you want to be cured of HIV, go and have sex with a virgin. There are also myths around companies are making a lot of money from treatment. So there is no way that the companies would lose all that money to a cure that will come and probably in one day, you don't need to go to the hospital several times. So there are all those kinds of different myths and misconceptions, and they're very deep in the community. So we need to do a lot of education for people to understand that the HIV cure is actually, something is being done around HIV cure. And if it comes, it will be accessible to everyone, including children, including pregnant women, you know, all types of people, because it cannot be a cure if it cannot cure everyone living with HIV. So as I wind up, I want to let you know that young people are less informed about HIV cure. They have that quest. They need to know more about what's happening around cure. But then those that are informed, including myself, we are excited, we are happy, but some of us feel like probably there's been some kind of slow movement towards achieving this cure, although we're excited with the advancements that are happening around treatment. Thank you so much. So I will proceed to the next slide and welcome my colleague and my friend, Martha, to lead us into her presentation. Thank you.
Thank you so much, Josephine. I hope you all can hear me. I'm hearing an echo. I'm so glad to be here. My name is Martha Ciccone Cameron, and I am an Elizabeth Glaser Pediatric AIDS Foundation ambassador. And I'm an ambassador because I am a woman living with HIV. I've been living with HIV since about 2003. And since that time, I've had two beautiful boys, just recently turned 11 and about to turn 13, who were born as a result of the amazing work that has been done in terms of research and prevention of mother-to-child transmission. My husband and my children are all HIV-free, and this makes this issue just very close and personal to me. I'm also the executive director of the International Community of Women Living with HIV, and I'll refer to that a little bit later on. But I wanted to raise 3 points today, and Josephine has even touched on one of them, but I'll still kind of expand on. The first one, I really believe that we need to celebrate PAVE and the fact that there is now cure research being done specifically on children and infants. I was invited 10 years ago, I reflected on this when I was invited to speak at this webinar in 2012 after the International AIDS Conference, to be on a community advisory board on one of the Martin Delaney boards called DARE. And the idea was for me to be there to advocate for research for pediatrics, as well as the inclusion of women. And I just remember how frustrating it was in those early years. A few years later in 2015, I heard about the Mississippi baby and even had the privilege of working with Debbie on some webinars and, just informing people, talking to people about what had happened. And here we are 10 years later talking about a Collaboratory that is specific to infants. So I really, really believe that is worth celebrating, and we will continue to do advocacy and support the work that is being done by EGPAF and PAVE for this issue. The second point I wanted to make, one of the things, especially having immigrated from Africa and also lived in the US, one of the biggest differences I've seen in our greetings is that in Africa, whenever you're greeted, somebody will say hello to you and they will immediately ask, how are the children? how are the children? And of course, a response to that will be, especially where I come from, very genuine. If the children are well, then the children are well. If the children are not well, you will say what is going on. And if you were to ask me today, ask me a global question about how are the children, my response to you would be, they are not well. And I think it's really goes without saying that even during this COVID pandemic, the last people to have been thought about were the children, and we're yet to see what impact will have happened on the children. Well, in addition to that, a lot of the trends that we've made in HIV were slightly reversed during this pandemic, including the fact that even here in the United States, there were a lot more children that ended up being born with HIV. And this is unnecessary, of course, in this time and age. And so when I think about the children and them always being the last people to be addressed or taken care of, I think what the repercussions of that are. And, Josephine alluded to different groups of children that, of young people and the way they are regarding the cure. Well, I would add to that, children that were had vertical transmission of HIV and are now in their teens and adolescents are struggling so much with mental health and just, navigating the transitions into adult care and navigating the world. Just in the past 2 months, I've had 2 calls to counsel 2 young people that have been suicidal because they just cannot manage their navigation of being HIV positive and transitioning into adulthood. The cure is needed for much more than just the adults. We need to think about these children that have had to live with this disease since they were born and the challenges that they're facing, and that this needs to be stopped. And the only way this can be stopped is when we have a cure for babies as well that are still being born with HIV as well. And then the final point that I wanted to make, and again, this has kind of like been touched on by Josephine, is just this idea of the knowledge, the scalability, and the sustainability of the cure. Very recently when the woman, it was announced that the woman was cured of HIV, I was asked to do an interview about it. And when I read up the details, and I've been very familiar with cure research because of my journey in being a CAB member and being connected to the research world. But I got to understand that even in this case, the woman had been cured because she had leukemia, and they had been able to cure both, similarly to the way it was done in Timothy Brown. And we know that this is not applicable to other people that do not have leukemia or cancer. We know that, for someone who's familiar, we know that there could be lessons learned that could be applied to other people. We know that this was exciting because she was a woman, where there's still only 11% of women participating in cure trials. We understand that she was a mixed race. So again, this was a teeny step in the right direction because we know that people of color have not been included in cure research. And so I will continue to advocate for the diversity and inclusion of a diverse kind of people being included in this cure research. Because as Josephine said, we know that the cure is not one size fits all. And we know that until it's applicable to everybody, it will not be celebrated. Just like this, this cure of this woman came around the community, and the community responded in, they kind of like looked at the information, but in the end, they were not that excited because it felt like it was not applicable to them. And so we want to continue to advocate for the diversity and inclusion of all kinds of people in cure trials. and as well as the education of the community. And I think even though Josephine talked about this in terms of young people, this actually applies to everybody. Doing a webinar does not inform the community, right? There needs to be an investment beforehand as you are planning this research into how community is going to be engaged, whether that means in terms of education, whether that means in support of the people who are participating, and whether that means for accountability in terms of ethics, especially when it comes to children. And so we really want to encourage you to include us as community, to educate us. And if you educate us, we will educate other people, and to continue ensuring that there's diversity and inclusion in the trials that you will continue to do. Otherwise, I will go back to my first statement and say, we are celebrating, I am absolutely celebrating PAVE, and that finally children are being focused on in terms of a pediatric cure. Thank you very much. And I'll hand it over back to Lynn.
Thank you, Martha. So folks, if you want to go on video, I can ask the questions that have been coming in. And maybe we can take the slide down and put people's faces up so they can be seen. So the first question I think is for Debbie, and it is that work focuses on HIV-1, but can these findings be potentially generalized to HIV-2?
I'll answer quickly, and that's a really terrific question, and we hadn't considered HIV-2 in the agenda, but know that there are differences in the pathogenesis of HIV-2 versus HIV-1. And so I would suggest you contact me. We could figure out how we can build a HIV-2 research program with this, because the broadly neutralizing antibodies responses may be different in HIV-2 versus HIV-1, which is something we can begin to assess.
Thank you, Debbie. I think Vincent is asking about P1115, the proof of concept study, and what have we learned so far?
So what we've learned is, we've gained information on pharmacokinetics of nevirapine. We're gaining information on raltegravir and VRC01 as part of very early treatment. In terms of the trials, we reported the 2-year outcomes at CROI this year. In P1115, which is distinct from the Botswana study and the leopard study in South Africa, ART interruption is planned for RV infants whose guardians and parents consent for ART interruption. And we have a biomarker profile of being HIV antibody negative. And I see that there's a question later about this, and low reservoir size, meaning no detectable circulating HIV infected cells. So what we estimate is 30% of the infants in P1115 reach that biomarker profile by 2 years of age to be considered eligible for ART interruption.
Thank you. This one, I think, Mark, is for you, and it's a question about Nabs and mother-to-child transmission.
No, thank you. So I think the first studies have been clearly for safety, and that has been achieved. And it's preparing the way for clinical trials, which, especially with the long-acting Nabs, may really prove excellent. One worries about resistance, and there already in impact, there are cocktail or combination studies that are in planning. So this is definitely the way to go, and we hope that this will actually happen. And of course, the convenience of a single injection lasting for 3 or 4 months is pretty amazing, and that's what our colleagues are working towards.
Thank you. Another question on Nabs are whether there might be a solution to enhance viral load suppression in high viral load patients or used for treatment interruption.
Yeah, no, thank you. And I think all of those are possible. I did briefly mention the Impact 2008 study where, in fact, that is being looked at with a single BNAB. it's been looked at, or be in the Telo trial has already used 2 BNAbs during treatment interruption for 24 weeks, and it was successful in almost half of the children. So there's a definite role in the future, but we're not there yet, but it's coming.
Josephine and Martha, just a question for you regarding enrollment into cure trials. You know, Josephine, do you think that you or other youth would be interested? And Martha, if it were your child, what would you do? Would you allow enrollment?
I'm not sure if Josephine wanted to answer, but Lynn, I would absolutely enroll my children. And the reason I would do this is because of the education and exposure I have received, because I understand that my quality of life today is because of research that has happened before me and because of the work, amazing work you all have done. And this is kind of like what I was requesting, like we need to be doing this education in the community and just encouraging and comforting mothers. For example, if we wanted to enroll babies in cure trial, to help them understand how safe, how very thoughtful, and how very protective the researchers are, and how this is important, you know, not just for now, but for people coming before us. So that is the education. I feel like I would absolutely, even cure trial, I would enroll tomorrow if something came up, but it's because of the exposure and education. And that's what we want to extend to the larger community.
Very good point. Josephine, your thoughts?
Thank you so much. Definitely, I think I'll agree with Martha. I would be happy, and I know that other young people would be happy if they've really gotten informed on what is cure research and what will be involved in that study. And for children in particular, I think this information goes to the parents, which I think even the P11, 15 really did, because for the infants, it's the mother that will decide whether the child gets enrolled or not. So it all comes back to information. Are they informed? The science that we know in cure research, can it be simplified in community language so that someone can easily understand if a study is coming up and probably they want to either enroll or not? So I think it comes back to information, but also among information, it comes to simplifying the science into community language.
Yeah.
Excellent, thank you. I have a question myself to Mark and to Debbie. So in the adult community, we talk about kick and kill and shock and kill, and they're using a variety of different agents, voronostat, rhodepsin, gene editing with CRISPR, and we're not talking about that with kids. And I'm just wondering what your thoughts are. Are we doing block and lock, or are we what are your thoughts?
That's a loaded question. So I think in terms of the kick and kill, the strategy that we're thinking about in PAVE is called surge and purge. And it's the idea of giving these innate immune enhancing agents, and it will be studied in the infant non-human primate model by Anne Sharudi and her group, is giving these agents at the time that treatment started. So instead of waiting until there's virus suppression and stable, more quiescent reservoirs, to see if we can surge and purge the reservoirs. With respect to what category of agents we feel that's the safest, you know, as you mentioned, vorinostat and romidepsin, those are epigenetic modifiers, and we don't think, with long-lasting effects, that we don't think we have enough information to use that in a pediatric population. But what we do have at our hand to study in older children and youth living with perinatal infection is IL-15 superagonist, which is a latency reversal, but it also is an NK enhancing, so natural killer cell enhancer, and also purges reservoirs, and gets into lymph nodes. And so with Immunity Bio, so part of PAVE is not allowed to do any clinical trials, but we can do preclinical work and ex vivo work to then provide the information needed for clinical trials. So we are working with Immunity Bio to begin studies of IL-15 superagonist to purge reservoirs in those living with long-standing infection. I do want to come quickly to the block unlock and the CRISPR-Cas9 approach. We've identified that it's harder to reactivate HIV from its latent state in perinatal infections compared to adult infections, suggesting a much deeper state of latency that we don't fully understand. And so maybe a block, instead of trying to reinforce the virus to reawaken, maybe a block unlock approach would be a good approach. And so there's one of the collaboratories, I think it's the HOPE collaboratories led by Melanie Ott, that's specifically focused on block unlock approaches. So as part of these collaboratories, we can work with the adult collaboratories and try to bring in those strategies for pediatric infections. Martha mentioned the case of the woman of mixed race that we reported on as part of IMPACT P1107, who received a CCR5 cord blood transplantation and is in remission. Martha, we didn't deem her cured because she's only 14 months off ART. And in that case, those CRISPR-Cas9, they seem futuristic, but it's a way to modify the CCR5 delta, or someone's own delta 30 CCR5 receptor to maybe get to that goal of genetic modification of target cells towards remission and cure.
Lynn, can I just add to that real quick? Because I remember the shock and kill was explained in lay language to me when I went to the Fred Hutch Center, and they said it's kind of like whack-a-mole, right? So virus pops up and you whack it, virus pops up and you whack it. And that was a great way to help the community understand what was going on. But on the other side of that is the idea that we have been working so hard to get people virally suppressed. So to help somebody understand, to be able to study this, we have to get you out of virus suppression. That's gonna require that, little bit extra education. So it's little things like that I just thought about when you talked about shock and kill, because I remembered whack-a-mole, that we would need to prepare the community to say, actually, we're gonna get you out of, that you're gonna get out of remission in order for this to be tried out.
Right, thank you. That's a good perspective. And also, I think, what Debbie pointed out is that some of these agents are not necessarily so safe. They're very experimental, and you should be reassured that the pediatric researchers are very carefully assessing what might be the safest for kids. And the idea that you would be using something to increase a patient's immune response to the virus and dealing with something like that rather than this, kick and kill, to me, I think should be reassuring. I think we are out of time. I'd really like to thank Debbie and Mark for the science, and Josephine and Martha particularly for your perspectives, which are really critical. And thank you everybody for joining. If we didn't get to your question, we will forward to the relevant person and see if we can get an answer. So thank you extremely for joining us, and bye.
Thank you.
Thank you so much.
Thanks. All right.